Hi. So now we're moving on to our second presentation, which will be done by Tomas from BIA Separations. And Tomas will be presenting on meeting demand for clinical production of mRNA. So I'd like to welcome him to stage.
Hello, thank you very much, Fatima, for introducing me. So I think the slides should be popping up. There we go. So thank you so much again. Good morning, good afternoon, everyone. Or I hope you've enjoyed a nice lunch or breakfast, wherever you're from. In this talk, I would like to focus maybe a little bit differently and specifically looking at some of the, let's say, challenges and demands that we need to meet to achieve or to reach the yields and the amounts of mRNA required for clinical production or clinical evaluations of different RNA therapeutic products, and specifically focus on some of the solutions that Sartorius has been developing and optimizing to help the field and to help manufacturers reach the amounts or the capacities required for clinical evaluations of messenger RNA therapeutics. And so we've heard in the previous talk a little bit about messenger RNA used for oncolytic applications. And historically, or let's say this has been one of the primary interests or primary motivations for development of mRNA therapeutics. Although we've seen in the past few years now, due to the COVID pandemic, that vaccine production of mRNA has, let's say, far surpassed and far accelerated the need, or let's say the amount of RNA that needs to be produced to meet the demand for the commercial use of messenger RNA therapeutics. But the trends that we saw in the past, so over the past decade, we've clearly seen an increasing trend in the number of clinical trials shown here on this slide. And so this trend is expected to continue, and especially when reaching late-stage clinical trials, this will require manufacturers and therapeutic companies to think about how to meet the demand to produce sufficient amounts of RNA for clinical trials. And we can take as an example what we've seen now with some of the commercially available vaccines for messenger RNA for COVID-19, and estimate approximately the amounts of messenger RNA that are needed to evaluate the efficacy of mRNA vaccines, let's say in a stage 2 or stage 3 clinical trials. And by taking some assumptions into account, we can reach a level of approximately 10 grams of mRNA, which would be the equivalent, again, with some assumptions on the productivity of IVT reactions, to approximately a 2-liter batch of IVT reaction. So the question we need to ask ourselves is, how can we achieve such yields? How can we purify this mRNA, and how can we minimize any losses that we see or that we obtain during the production and during the purification process? And to do so, we need to understand a little bit about, on one side, the structure of mRNA, and on the other side, about the actual production process that we use for mRNA manufacturing. Messenger RNA, as we probably all know, is a single-stranded RNA molecule. We can, broadly speaking, distinguish two different classes of RNA therapeutics or RNA vaccines, one of them being traditional messenger RNA, which will range in size between, let's say, 1,000 to 5,000 nucleotides. And on the other side, the so-called self-amplified mRNA molecules, which are messenger RNA molecules containing additional replication machinery or encoding additional replication machinery, which allows the cells to actually produce copies of messenger RNA, therefore extending or lowering the dose required or extending the expression of the protein or of the target protein. If we look at the structure of mRNA, we see that it's composed of three primary, let's say, parts. The middle one, and potentially the most important one, is the genome sequence, flanked or with each side covered by two untranslated regions. And this part is the one which dictates or is actually producing, encoding the protein or the target molecule that we want to produce in our cells. On both of the sides, so on the front end, on the 5' end, we have a 5' cap, which is actually a modified nucleotide. And this 5' cap is one of the elements which is required for the biological expression of proteins inside of the cells. And on the other side, on the 3' end, we have the polyadenylated tail. And just in the same way as the 5' cap, the polyadenylated tail is required for our cells to produce or to translate the mRNA into proteins. So all of these structural components of messenger RNA have to be added during the production process onto our final product, so onto the drug substance or drug product. And therefore, we need to understand how messenger RNA is produced. The production process of mRNA starts typically with a linear plasmid or linear DNA template, which is normally acquired or produced in the form of a plasmid DNA, which is linearized and then used in an in vitro transcriptional reaction. This in vitro transcriptional reaction combines our linear DNA templates, nucleotides, enzymes, and other reagents which are required to produce or to transcribe the DNA into the mRNA product. Now, it's important to understand that the 5' cap and the poly tail are not necessarily added to the mRNA product at this stage. So additional reactions can be needed if these two elements are not added during, or let's say, co-transcriptionally. But in most cases that we see, the polyadenylated tail is typically encoded into the plasmid DNA, and so it is already transcribed onto the mRNA product, while the 5' cap is typically added either with capping analogs or co-transcriptionally during the IVT reaction, or as a secondary reaction, enzymatic reaction, for example, with vaccinia capping. Now, to achieve high yield and high productivity of our IVT reaction, we need to make sure that our understanding of this process is as high as possible. And similarly, it's just as important to achieve high recoveries and high productivity during the purification process. So the tools and the methods that we use both for analytics, so for understanding the IVT reaction, and later on for purification need to meet or need to be suitable for analysis or for purification of large biomolecules like messenger RNA. And this is the first, let's say, critical or key point. Messenger RNA is a very large biomolecule. If we compare it to traditional biologics, like, for example, monoclonal antibodies, which are by themselves considered large biomolecules, messenger RNA is several times the size, both in terms of molecular weight and in terms of physical size. And this imposes a certain number of restrictions when it comes to selecting suitable analytical and purification solutions. One of the more frequently used methods, both in quality control, so analytics, and in purification in industry, is chromatography. And when selecting a chromatographic medium, we need to make sure that the stationary phase, the matrix that we're using, provides suitable properties, both in terms of accessibility of the surface area and low shear environment, as well as easy scalability and high capacity to be able to scale up from a bench scale production of mRNA to a clinical production or commercial manufacturing. And within this context, the monolith, or the structure of the monolith, is relatively unique in terms of chromatographic media available for purification of mRNA. The monolith itself is produced as one single piece of polymer, permeated or filled with channels. And all of these channels are relatively large, providing sufficient accessible surface area for messenger RNA and eliminating the need for diffusive processes, which are the guiding force behind mass transfer for porous particle chromatography. So the laminar flow inside of the monolith and the convective mass transfer give us two critical advantages, which are very important for purification and for analytics. On the purification side, one of the more important driving forces is the flow-independent binding capacity that can be achieved with the monolith. If we take a comparison shown here between a porous particle column and a convective monolithic cation exchanger used for capture of, in this case, an antibody, we can see that increasing the flow rate on the porous particle column leads to a loss in the binding capacity. And this is where the concept of residence time becomes very important. With porous particle media, a sufficient residence time is needed to achieve adequate or suitable binding capacity and suitable resolution of our chromatographic medium. With a monolith or with a convective chromatographic medium where the performance is independent of the flow rate, we are no longer bound, or we're no longer in need to understand the residence time in our process, and we can adjust the flow rates as needed. This leads to an increase in the productivity and in the speed of the production of mRNA. And the same is true for analytical applications, where flow rate independent performance is critical to achieve high throughput analytics and basically a very rapid analytical assay. So this is on the side of the stationary phase. Without going into any more detail, we then need to look at the surface modification of these stationary phases, of these monoliths, and the surface chemistry of the chromatographic column is the one which dictates the resolving power or the separation capability between the different components in our samples. And so we're looking here at three different analytical chemistries which Sartorius provides in the portfolio, and how each of these is used for different purposes in the context of mRNA. So the first one is the CIMac Prima S column. CIMac Prima S is a multimodal anion exchanger which allows us within a 3 to 3.5 minute gradient to separate from each other the capping reagent, individual nucleotides, plasmid DNA, and messenger RNA. And so this allows us to evaluate and understand our IVT reaction in a very short assay in very high detail. The second chromatographic chemistry that we're looking at here is oligo-DT, which is an affinity chromatographic resin which binds the polyadenylated tail. And so all of the RNA species, or all of the species which are polyadenylated, will bind onto the oligo-DT ligand, whereas any non-polyadenylated species will be in the flow-through. And you can imagine that this type of analytical method can be used to very rapidly assess the titer or the amount of polyadenylated RNA in our IVT reactions. But it can also give us a more deep insight into the structural integrity of the mRNA over time. For example, we can look at degradation products of our mRNA molecule, where the fragment which still contains the poly-A tail will bind onto the oligo-DT column, whereas the damaged fragment, or the fragment without the poly-A tail, will go in the flow-through. So this is a very good rapid analytical assay to look at stability, for example, of mRNA. And the third approach shown here is reverse phase chromatography, or ion pair-reverse phase chromatography with CIMac polystyrene divinyl benzene. And this reverse phase approach gives the highest possible resolution for different sizes or different fragments of messenger RNA. So it gives us a very good resolving power, for example, shown in this case for an RNA ladder. But it also has the highest degree of separation, or the highest ability to separate double-strand RNA contaminants from our single-strand or our mRNA product. And we can use all of these in different formats. So we can use it either as an analytical column with an HPLC or UPLC system, or we can use them in a high throughput format like a 96 well plate, which gives us, let's say, even wider optimization and analytical opportunities. I would like to focus, though, to the point that I've introduced earlier, which is optimization of the IVT reaction. Now, to achieve the highest productivity during the IVT process and to shorten the production times, both in terms of mRNA production, but also development of the production processes for mRNA, we need to have an analytical assay which is rapid enough to meet or to match the speed of the IVT reaction. So if we compare IVT to the IVT reaction to a traditional fermentation process, which can take days to weeks, the IVT reaction typically reaches its time course or its endpoint within 2 to 3 hours of reaction time. And so we need to have analytical methods which can give us detailed information on these, on what's happening in the IVT reaction in real time. And most traditional laboratory methods or biological methods cannot achieve such short analysis times. And for this purpose, we've implemented then this multimodal Prima-S column, which, as mentioned before, gives us the ability to separate the different nucleotides from each other, as well as the capping reagents, the plasmid DNA, and the mRNA. And all of these can then be quantified and analyzed over time to understand how our IVT reaction is progressing. Just to give an example of how this can be used, we can take a sample from the IVT reaction every 10, 15, 30 minutes, quench the sample, analyze it, and integrate the peaks that we obtain on the chromatogram, which we can then plot on a time axis to understand how our nucleotides are being consumed and how our mRNA is being produced. Now, with this type of information, we can, of course, expand the scope of applicability, and we can look at how temperature affects the production of mRNA. We can look at how concentration of nucleotides or concentration of capping reagent affects the productivity of our IVT reaction. And with all of this information, optimize the parameters which go into the IVT reaction itself. Now, if we take it even a step further, we can perform additional sample manipulation, so additional material handling, which allows us to estimate or to measure the capping efficiency with the help of the same assay on the Prima-S column. Specifically, we take a sample of the IVT reaction and treat it with a combination of 5 polyphosphatase and terminator exonuclease. The combination of these enzymes will selectively degrade uncapped messenger RNA. And so we can take this same Prima-S assay and analyze a control sample and the sample which was treated by the enzymes to understand how much of our messenger RNA in our reaction is capped versus how much of it is uncapped. And this is shown here as an example where we look at the capping analysis of messenger RNA for a few different constructs by splitting them in half, taking 2 aliquots, analyzing one on the Prima-S column, treating the other one with enzymes and analyzing it on the Prima-S column. And the relative surface area between the 2 peaks gives us a value for the capping efficiency of our reaction. Now, development of analytical methods can be time consuming. So within Sartorius and BIA separations, we have compiled all of the hardware and all of the software together to provide ready-to-use solutions for mRNA and plasmid DNA analytics, and specifically an HPLC system with all of the hardware components, as well as all of the analytical methods already built in and validated so that they can be directly implemented onto the production workflow for mRNA. Now, once we've optimized our analytical approach, and once we have our IVT reaction tuned down to have the highest possible productivity, then we can start to think about the purification approach for our mRNA product. And it's worth looking at some of the typical purification methods employed for mRNA at different stages in let's say, in the production or development of an mRNA therapeutic. So on one side, we have precipitation methods like precipitation with lithium chloride or solvent precipitation, which are very often employed in small-scale environment in laboratories. But they bring along a few different challenges, specifically a challenge in terms of scalability and toxicity of the materials used, but also in terms of the stability of the RNA products, as I will show later on. So this approach is suitable maybe at laboratory scale, but for commercial therapeutic production, it will not be used. And it's worth implementing a scalable production approach from the very beginning to facilitate the transition into clinic. Now, another purification approach which is frequently used for mRNA is tangential flow filtration, which is a fantastic method that provides scalable concentration and purification in one single step. The drawback of this approach is that separation or purification is achieved based on one single parameter at a time, and specifically the cutoff size or the molecular weight alone. So we're not looking at selectivity based on the nature and properties of the contaminants, and so the selectivity of this method will be lower compared to something like chromatography. Now, the same way we saw with analytics earlier on, chromatography provides a very high degree of selectivity by selecting the right type of chromatographic resin, but it also provides excellent scalability, which allows us to transition into clinic and then into commercial manufacturing relatively easily. The challenges in terms of analytics that we saw earlier also apply to purification. Specifically, we're looking at the size of mRNA, the shear sensitivity, and the stability of RNA product, but then also the lack of standardization of IVT processes. As I've mentioned, we're looking on one side at processes which can introduce, for example, the 5' cap within the IVT reaction, and other processes which do not. And for this reason, one single platform does not necessarily apply to all mRNA processes, but a purification toolbox with a selection of analytical columns, of purification columns, can provide the right amount of flexibility. So within Sartorius, and I will focus here on the capture step primarily, we provide affinity chromatographic capture with oligo-DT and multimodal chromatographic capture with Prima S, and I will go into each of these a little bit later on and discuss them in a little bit more detail. In terms of polishing, again, this depends on the nature of the contaminants which are left in our sample and need to be removed after we've performed the initial capture step. So the oligo-DT affinity chromatography allows capture of mRNA directly from the IVT reaction with very high capacity and a very short implementation time. With the Monolith, which are caustic stable, the columns can be sanitized, cleaned, and reused for consecutive production campaigns, which reduce the overall cost of the chromatographic medium. The important aspect to look at is that our product, our eluted mRNA, needs to be stable and needs to be pure or free of contaminants after the purification step. And so we've done we've performed a case study where we looked at the mRNA stability, or let's say the thermal stability of mRNA, which was purified under different conditions. And specifically, we've taken an IVT control sample, which was an IVT reaction, an oligo-DT purified material, and a precipitation purified material using a commercial RNA extraction kit. And we've exposed the purified material to different temperatures and analyzed the stability over time. The result is shown here on the agar gel and the bioanalyzer data, where we can see on the agar gel, the material which was purified by precipitation showed a significant or a complete loss of our RNA product when exposed to 37 degrees for 28 days, whereas both the control sample and the oligo-DT purified material showed relative stability under these conditions. The same is shown in the bioanalyzer data, where our RNA extraction kit resulted in complete loss of our RNA product in 28 days, whereas both IVT control sample and oligo-DT purified were stable under the same or relatively stable under the same conditions. So the methods which are used for laboratory scale purification may not even be suitable for this purpose if the messenger RNA will be stored for a prolonged period of time. Now, what happens if our messenger RNA does not have a polyadenylated tail after the IVT reaction? So in that case, an oligo-DT purification approach cannot be employed, since we don't have a poly-A tail to bind onto the oligo-DT column. And I've described before this Prima-S multimodal chemistry that we use for the analytical scale, where the same thing can be translated into a larger format. And this is precisely what we do, with one major difference. Instead of using a pyrophosphate gradient, we employed a pH gradient elution. And by doing so, we can achieve practically the same separation that we saw before, where we separate all of the nucleotides, reagents in the IVT reaction from the plasmid and from the messenger RNA. And we perform this elution in an ascending pH gradient, which gives us our RNA product. Now, since elution takes place in a pH above 10, we've then, of course, had to look at the stability of the mRNA products once more to ensure that the purification method we're using is not damaging our mRNA. And we've performed a very similar study to the one we saw before for the oligo-DT, where we've exposed our mRNA to different temperatures for a specific duration of time. And we've seen that when the Prima-S eluate is immediately neutralized upon elution, the stability of mRNA is very high, and we don't see any degradation at room temperature, even at 28 days after purification. So this is an alternative approach which can be used for capture and purification of mRNA directly from the IVT reaction with very minimal sample preparation. So if we look back to one of the opening slides, where we looked at the requirements or the needs for clinical manufacturing of messenger RNA, we can look again at the different scales needed, both in process development, clinical production, and manufacturing scale, and look at, let's say, the capacity or the amount of mRNA that we purify and what size of chromatographic columns we would need for each of these processes. And so we can see that, let's say, with the smaller scale, with the 1 milliliter scale, we can purify 6 to 8 milligrams of mRNA per run in, let's say, in the capture step. And if we translate this onto a purification of 10 grams of mRNA, we would typically need to use around 1 or 2 800 milliliter columns, whether we're doing reusing, so recycling the column, or we're employing 2 separate columns. And this process can then be scaled up to manufacturing scale to produce several multigrams of mRNA, even several hundred grams of mRNA per run in the larger scale. So some of the key take-home messages here are that, meeting production of mRNA and specifically meeting the timelines to reach the cleaning with messenger RNA products requires, first of all, development of suitable rapid analytical methods to be able to optimize and understand our IVT reaction. And in the next step, developing purification methods which allow us to obtain high purity and high recovery of messenger RNA. Now, just to finish off, historically, Sartorius in Slovenia also works on process development and provides development of both analytical methods and purification methods with tech transfer to different customer sites. So outsourcing is also one of the options that can help a therapeutic company reach the goals or reach the clinic within a shorter amount of time. And so I'd like to close off with this and maybe just mention that you're very welcome to reach out for a copy of the purification handbook for plasmid and mRNA. And thank you very much, and I welcome any questions.
Thank you, Thomas. So let's see if we've got any questions. I'll give it a minute just in case there's a lag or anything. Lovely. So we've got a question, as you can see in the chat, from that's from Yogesh. Yeah, if you could read the question aloud and answer it, please.
Okay, so we have a great question. So does the system that we've presented here allow purification of NTPs? And so maybe just to understand the context, so whether are we looking to isolate NTPs and purify NTPs in the production process, or separate NTPs, nucleotides, from our mRNA product? Now, the answer is the mRNA can be clearly and easily separated from the nucleotides that are used during the IVT process. We've seen this in the example of the Prima S column, where the nucleotides are actually individually visible on the chromatogram. And it provides, let's say, removal of all of these nucleotides from our IVT material. I see that we have another question. So there is a question about lead time. So what is the lead time for the different purification scale columns, and whether COVID pandemic has affected the timelines? This is actually a fantastic question, because we've seen the field has struggled a lot to meet demand during the COVID pandemic. In the case of the Monolith, we've actually increased several-fold the production capacity for mRNA just before the pandemic, which in turn allowed us to meet all of the demand that we had during production of these mRNA vaccines, but also to shorten our timelines of all scales of purification products. And I believe the longest timelines, or the longest lead times, are normally maximum of 6 weeks for any product.
Amazing. Yeah, I think that's everything in terms of questions then. I can't see any more. Yeah, thank you, Thomas, so much again for your presentation, and really appreciate it. And the questions previously from Volker's presentation that we unfortunately missed, we will be emailing those questions to Volker and letting the people who ask those questions know what the answers are. But yeah, now we have a half-hour break, and then we will continue on with our next presentations. But again, thank you so much, Thomas. Really appreciate it.
Thank you, Fatima. Thank you, everyone.
Thank you.
Take care.